Great, thank you so much to the organisers for inviting me. It's great to be back in Bielefeld. I've got a lot of friends here and collaborators, so it's a real privilege to be here. This is my career in one slide, if you like. I started my PhD working on this animal in the top left, the banded mongoose, and I'm still working on banded mongooses now. I often get asked, surely you've found everything out you want to know about banded mongooses, but I'll hope to convince you that there's still plenty to work on. I then moved to a postdoc working on paper wasps. Brilliant animal, harder to get people interested in unless they're scientists, like my mum, for example, wasn't keen on paper wasps. But yeah, just a phenomenal animal to try and understand conflict and cooperation in animal societies. I then got interested in life history evolution, particularly in humans, and the evolution of the menopause, the female menopause in humans in particular, and that led me on to working on killer whales as part of a project where we tried to test some of these theoretical models to explain the evolution of menopause in one of the only other systems where you can actually get the data to test these ideas. And I think when I started working on these cooperative breeders, they're all cooperative breeders, I was really kind of interested in this long-standing puzzle about the evolution of cooperation. But at the time that I started my PhD, this book on the right came out in 1995 by Maynard Smith and Seth Mary. And I also read this fantastic book by Leo Buss, The Evolution of Individuality. And this kind of placed the whole problem, the behavioral ecological problem of the evolution of cooperation in a much sort of grander setting. And that I found quite inspiring to think that the evolution of cooperation might be this repeating motif in the evolution of biological complexity. That was the kind of thesis put forward in this kind of major transitions framework. And the idea is that life has become more complex through a series of these cooperative transitions where independently reproducing units have come together to form cooperative teams and started to reproduce as teams. These form the basis of the next level. It kind of forms a social ratchet, if you like, in complexity that can form the basis of the next level of complexity. So here's a little cartoon of how that process is thought to occur. So the formation of from selfish units, you form these kind of social groups, and through this process of conflict suppression and elaboration and specialization and division of labor, you get the formation of new units. There's also something else that happens during those evolutionary transitions, which is that we have to change the way that we think about fitness. Instead of thinking about fitness at the level of these units that are making up these kind of intermediate social groups in the middle there, we have to think about fitness shifting to the success of collectives at producing new collectives. So this is a shift from what I'm calling type 1 to type 2 fitness. These are some of the major transitions that you'll be familiar with. The origin of life is thought to have involved this kind of cooperative transition, the evolution of complex cells, eukaryotes, through symbiogenesis. And this, starting about 100 million years ago, the first transitions from individual organisms, what we'd recognize as an individual organism, to these kind of eusocial societies where it makes more sense really to think about the colony as the level of organismality. And the same kind of principles have been applied to try and understand the evolution of human societies, though it's probably less clear how some of these ideas, particularly type 1, type 2 fitness, whether they really map on well to understanding the evolution of the kind of elaboration and increase in scale in human cooperation. And also after Athena's talk, you know, it's interesting to think how you might apply major transitions theory to the evolution of AI. So I think that I've been interested in this transition because cooperative breeders occupy this muddy middle ground. They're not kind of fully integrated societies. Each individual retains the ability to reproduce itself. So they're a fantastic kind of crucible to think about the kind of forces of conflict and cooperation that are boiling away there and helping to shape these societies. I think I started out for much of my career interested in the evolution of help and division of labor, how conflict is suppressed. And more latterly, I've become interested in this transition from one type of fitness to another, because some of these cooperative breeders exhibit both at the same time. But what I want to, there's other processes that occur as well during these transitions, which I want to focus on in this talk, and that's the evolution of a new group identity. There's a new identity that has to emerge as a process of these transitions to higher levels of social organization. And also those societies make decisions. They evolve a level of collective agency or group agency. So I'm going to focus a little bit on how group identity evolves in biological systems, and also at the end maybe make some speculations about how that relates to group agency. So that's the plan for my talk. I'll go through these 4 stages, and you'll see a lot of this animal, which is my kind of go-to study system to try and address some of these questions. But first, a little quick tour of what I mean by social identity. So I'm using social identity here to mean the kind of perceived membership of a social unit, which influences how an individual acts and how it's treated by others. And I think there are 3 kind of main categories in biological systems which seem most relevant. There's a kind of kin identity, so discrimination of which individuals are kin and which are not. There's, in some systems, discrimination of individual identity, where who you are matters in social interaction. And there's, in other systems, discrimination of group identity, discrimination of which group you belong to. Particularly in the context of intergroup conflict and being able to discriminate between insiders and outsiders. There's a couple of points there. I think social identity is an evolved feature of certain systems. It's not a prerequisite of social interaction, and no identity is required in many systems. And secondly, I've kind of made kin identity bigger there in this Venn diagram because that's kind of ancestral. I think microbes have kin identity. I'm not sure that they have individual identity. It seems kind of to apply to a very wide range of species. But as I say, you know, many systems don't require any kind of identity. I used to study these kind of chironomid midges when I was my first paper was on chironomid midges, which are swarms of males waiting to intercept females coming into swarms that you see quite often in over African lakes, Great Lakes like Lake Victoria here, or mussel beds or broadcast spawners, these types of scramble competition involving one-off competitions. There's no requirement for any kind of identity and no, as far as I'm aware, no discrimination of any kind of identity. Kin identity is much more widespread and occurs in a vast number of organisms where, and this is the kind of one of the 2 major processes that can lead to the evolution of traits by kin selection proposed by Hamilton. If you can discriminate those that share genes identical by descent, then you can create the conditions for altruism to spread in populations. In other systems, some cooperative, some not, you also have the evolution of individual identities, famously in the facial patterns of these paper wasps, which are one of the few insects that exhibit individual recognition, but also in other situations where you need to keep track of individuals, such as in dominance contests, dominance hierarchies, in these vampire bats where there's tracking of individual donations in food resources, or in these kind of mutualistic systems where there appears to be evidence in cleaner fish for some level of individual recognition. And then the final level is this group identity, where I think applies most to where you've got genetically heterogeneous groups, but nevertheless shared interests. So I've kind of placed a sort of barrier around a collection of individuals there, such as occurs in extended families and where there's intergroup alliances and so on. And these can be discriminated on the basis of shared phenotypic traits, which I'm going to go on to talk about a little bit more. And I think immune systems as well can be understood as one type of group identity recognition system. I'm going to talk about two routes to group identity that are not exclusive and may complement each other. The first is to suppress individual identity or markers of kinship, which has sometimes been called the veil of ignorance in biology, in reference to similar ideas in philosophy and economics. And the second is to actively form a new group identity by which you can discriminate insiders from outsiders in your social group. So the veil of ignorance is a term that, as many of you will know, is borrowed from John Rawls' very influential book in the 1970s called A Theory of Justice, which itself was a kind of built on earlier ideas in game theoretical models of incomplete information pioneered by John Hassani, which asked what rules of resource allocation, what division of resources would a rational decision maker choose if they were ignorant of the personal gains that would result from this allocation. Rawls used that as a kind of principle by which societies might rationally choose arrangements which were just. In biology, that term has been borrowed to describe situations where uncertainty or ignorance of genetic relatedness or the payoffs of social interaction can actually promote cooperation within social groups. And this is a view that's been advocated by, or an idea that's been written about by Samir Okasha and Joan Strassman and Dave Queller. So here's some examples of systems to which this veil of ignorance concept has been applied. At the cellular level, or the intragenomic level, the veil of ignorance has been used to describe how uncertainty about identity during the process of meiosis can actually prevent and suppress intragenomic conflict. So meiosis is famously usually a fair raffle where chromosomal number is halved and each chromosomal homologue has an equal chance of getting into the gamete. And that uncertainty about the kind of fixed transmission rate of both of these alleles or both genes on a homologous pair of chromosomes means that the best thing a gene can do to increase its representation in the next generation is to increase the fitness of the organism, both chromosomal pairs. There's also processes early in development like the stripping of parent of origin markers from chromosomes that occurs during germ cell formation in the developing embryo, which prevents there being any markers of genetic lineage within the genome, which could otherwise break out into kind of nepotistic conflict within the genome. In honeybee patrolines, queens mate with multiple males, but workers don't discriminate between half-sisters and full sisters. They do discriminate between worker-laid and queen-laid eggs, but cues to identity and relatedness among the offspring of queens are suppressed. And the same is true in multiple queen ants, where larvae are moved around and mixed up between piles, essentially drawing a veil of ignorance over relatedness within these piles of brood. I'm going to give you another example of this, which is from my own research on banded mongooses, where this species, one of the first things I found out about it is that they exhibit quite a remarkable degree of birth synchrony, which seems to mix up cues to identity in communal litters. This is our study site. It's this beautiful peninsula in the middle of lake, juts out into Lake Edward in western Uganda. It's on the border with the Democratic Republic of Congo. And I set up this project for my PhD research in 1995 with this chap here, who's Francis Mwangusha, And Francis is still the field manager of the project. So we've been together for 30 years. This is his beautiful family on the right. And I've had the privilege of getting to know an amazing team of Ugandan researchers who are just incredible. Anyone who works on that project knows just how fundamental that team is to the success of everything that we've managed to do in the Banded Mongoose project. It starts from kind of small beginnings. It's become quite a big project with lots of fantastic collaborators who I really enjoy working with, including collaborators here at Bielefeld. So what do banded mongooses do? This is what, the first thing in the morning, they wake up about 7 o'clock, nice respectable time to get up, and then they head off on a foraging trip where they dig up beetles and millipedes and things. And then it gets to about 11 o'clock, 11:30, and it gets too hot, and they just sort of go under a bush and lie around for 4 hours. And then about 4 o'clock in the afternoon, they emerge and do another foraging trip and kind of wind their way back to the den that they're going to sleep in that night. We have 3 or 4 groups usually at one time who are extremely habituated. So out of 10 or 12 groups that we're monitoring, there's usually 3 or 4 or 5 that will let us get this close, where we can really collect some detailed information. We trap the whole population, we visit them on a daily basis, and they kind of weigh themselves, which is handy. And we've got a very large database, which over the years, and we've added really kind of some fantastic cutting edge techniques to try and understand individual relationships and physiological effects and genetic relationships within this population. So Hazel Nichols has led the genetic work and working with Joe Hoffman here at Bielefeld. Faye Thompson has been really, really important putting the life history data together. I do want to kind of put a shout out to my current postdoc, Laura Labarge, who's got this amazing task of trying to set up a reverse GPS tracking system and dealing with just amazing logistical challenges right now in the field in Uganda and solving them. We've also started using drones and automated tracking, which is part of our kind of current ERC grant to try and use these techniques to understand leadership, cohesion and dynamics within groups. But I'm going to talk about birth synchrony. This is, like I say, this was the first thing that I discovered in doing my PhD. You know, you'd follow a group and there'd be 4 or 5 females who are heavily pregnant one day, and then you'd go back the next day. The males come out of the den at the normal time, but the females stay behind, and then about 10:30, the females emerge, and they were all slim. We realized that they'd given birth together on the same night, same morning, as it turns out. And this wasn't just a one-off, this is kind of the norm. So on average, around about 4 or 5 females give birth synchronously on each breeding attempt. We've had up to 12. Once 12 females synchronized birth to the same day. And the mothers suckle those offspring without any discrimination at all of kinship. So they'll kind of be looking around and pups will move from one female to another. So there's no apparent discrimination of maternity within these litters. And after the pups emerge, they do another weird thing, which is that they form one-to-one relationships with particular adult helpers that we call escorts. And those escort pup pairs are also no more closely related than you'd expect by chance. There's no apparent discrimination of kinship in the formation of these kind of teacher-student relationships. And those escorts will care for the offspring. They teach them foraging tricks, and so on. We know that asynchrony is really bad. About 30% of litters are asynchronous naturally, and in those litters, many fewer pups survive, and they tend to die very quickly. Particularly the females who give birth early, their pups usually die straight away. And lots of anecdotal evidence suggests that those pups are killed by other females that are still pregnant. This kind of effective birth order on infanticide is actually quite a general pattern that you see in lots of cooperative breeders. where females seem to be infanticidal until they produce their own offspring. But I did an experiment starting in 2007. I got a 3-year grant, and it took 7 years to do the experiment, to try and manipulate the distribution of reproduction among these females, to try and understand the costs and benefits of sharing reproduction, and also the kind of behavioral mechanisms that were used to enforce control over reproduction. So for some of these breeding attempts, we suppressed, this is before a use of short-acting contraceptive injections. This is before, this is where we suppressed all the subs for one breeding attempt. So these are dominant females, and that was a sub-females. And then that's after. So you can see we were successful at kind of switching off these subordinate females for one breeding attempt. Here, I kind of turned them into meerkats. So I was trying to kind of take the oldest female and just leave her as a breeder. And here we suppressed all the subordinates. This one, I really thought that they would do much better, because I thought you're removing competition from these other females, that the survival of those offspring will be better, that they'll have this more efficient division of labor, as you find in meerkats. But it didn't turn out like that. We got these very strong patterns of litter death in the first week, where there was no real effect when we switched off for female subordinates. But if we switched off any of those dominant females, then the litter died very quickly. In fact, I kind of stopped this experiment after 8 breeding attempts, because it was clear that there was a very strong pattern there, an unexpected pattern. So for ethical reasons, we kind of curtailed the experiment. So I think this was quite difficult to get experimental evidence that dominant females do kill litters which don't contain their own young, either because they're still pregnant or because they haven't contributed to that communal litter. And that fits with the idea that birth synchrony, if you can synchronize birth to the same day as dominant females, then there's this mixing of cues to maternity, which leads to no infanticide, and allows subordinates in particular to have surviving offspring. Well, what are the consequences of this veil of ignorance? And I was kind of interested in this prediction of roles, that from behind the veil, this was a verbal model of roles, but from behind this veil of ignorance, what rational actors should do is allocate resources in a way that provides maximum benefit for the least well-off, which he called the maxi-min principle. And that would typically reduce inequality. So we did an experiment where we fed half the females in each group during pregnancy, which increased inequality. Those females, we left the other half of pregnant females as controls within the group. They all gave birth on the same day, and then we followed what the patterns of care were. This was an experiment led by Harry Marshall and Robert Businji, who has developed an amazing way to feed females selectively. It's a very difficult thing to do. So we were successful at creating inequalities. So if we fed females in the latter half of their pregnancy, they produced bigger pups. But then, typical mongoose style, they did something really weird, which was that those fed mothers, who were heavier and in better condition, went and spent most of their time escorting the smallest pups in the litter, who were the control pups, the pups of the non-fed females. And that resulted in a leveling up of pup weight. So this is inequality on the right. This is before the period of postnatal care. As a consequence of this strange pattern of postnatal care, they were successful in reducing inequality in these litters. Whereas litters that we didn't manipulate, there was no change in the level of inequality. So birth synchrony in banded mongooses does seem to remove cues to kinship. It prevents infanticide, and it promotes fairness in the distribution of resources in a way that's kind of consistent with this maxi-min principle, that care is allocated to provide the greatest benefit to the most needy. So this is what ChatGPT makes of these kind of Marxist mongooses. And we showed theoretically that this maxi-min principle can actually, which has been debated quite a lot in economic game theory, and Harsanyi wasn't very keen on, that the maxi-min principle can be adaptive when the stakes are very high, and the best thing is to avoid being in that worst off position. Okay, I'm going to quickly, this is much quicker, talk about group identity because this is work in progress. I want to talk about this kind of second process, not the stripping away of individual identity, but the formation of group identity. And the idea that I think it's particularly relevant and particularly conspicuous when in the context of extreme intergroup conflict, which is another feature of the mongoose system. So these labels of group identity can take many forms. I think in immune systems, in some microbes, they take the form of surface proteins. In many social insects, CHCs are used as markers of group identity, or other volatiles in mammals, and in humans, dialects, symbolic markers, languages, and so on. So existing theory says that good group identity labels should be traits that are shared by members of the same group and not shared by members of different groups. That makes sense. They can be genetically determined. A lot of the insect theoretical models for social insects assume that they're genetically determined, or they can be learned or environmentally acquired, but they're generally thought to be kind of self-controlled. And the idea is that, if you've got like this actor on the right who's like an ant guarding the entrance of a nest or something, it's got to tell whether to let this ant in. The recipient kind of comes up with a label, the actor compares it with a template that it's got, a kind of cognitive representation of a template perhaps, and decides on its optimal response. And the difference between the template and the label can be used to make a discriminating decision. So you can set your discrimination threshold somewhere along this axis of the difference between the actor's template and the recipient's label. If you kind of increase your threshold and allow more difference, then you run a greater risk of letting in individuals that don't match your group. If you set it too stringently, then you run the risk of excluding those members of your own group in error. So that's fine, but in current theory, these distributions of group identity are fixed, whereas in many animals, I think, they're the result of behavior. I want to kind of illustrate that by showing you some videos of mongooses quickly. This is mongooses kind of lining up against each other in what we call battle lines. And you'll see that this group at the bottom here, I want you to watch for sort of rubbing against each other, which we call allomarking. So there's a lot of hopping about on top, and they're dragging their anal pouch over the fur of each other. It's chaos, by the way. And in the heat of battle, I think that the smell of animals is used as a really quick check of who they are, rather than visual cues. I mean, visual cues and so on, I'm sure, contribute to identity in mongooses too, but in the heat of battle, smell matters.
There's one head, and the other side is 11.
I'm going to show you this at full speed, and then I'll walk you through a slow motion version of it.
Organizing to start a fight again.
This an animal here, there, is going to get caught.
Always.
The group coming back to rescue it. Apologies for the sound there. I'm going to give you a sort of action replay of that. These are our two main protagonists here. This guy is going to come and be the rescuer. This one is the one that's going to be overconfident and make a miscalculation and end up caught. So at first, he chases off this one at the bottom, comes back around, and now he's overconfident. He leads the charge, and he's going to come face to face. These will flee. He's going to come face to face with a mongoose that doesn't flinch. which is quite interesting. So there he's going forward. There's the guy who's not flinching. He thinks, right, I'll have you. And here's his henchman. These are males, by the way. Here's the henchman coming, and now he's caught. Trouble. And mongooses can get really seriously injured and killed in these kind of fights. Here's the other protagonist. He's coming back, and he's going to kind of, funny to watch what he does. He comes and he's interested in trying to get a sniff of who it is that has been caught. He came back has brought his other group mates out of hiding. He gets entangled in something else over here. But they're going straight for the smell. I suspect they're possibly kind of biting and so on. Anyway, I'm going to speed up a little bit. One other video, the other time that they do a lot of allo marking is when, for some reason, if you pour water next to them, they just do this crazy thing. They don't do it when elephants urinate next to them. A lot of allo marking. Interesting. So we've got a hypothesis that it's really kind of, you know, if all your conflict is kind of within group, then it pays to advertise your individuality. But if most of your conflict is between group, then it can pay to homogenize these labels. And the thing that I'm interested in is that the mongooses are flipping between these states. You know, they're squabbling amongst each other. If another group appears, then they go and start rubbing each other and go into battle. But there's kind of movement. This is not kind of fixed template distributions. These are kind of behaviorally mediated. And with Rufus Johnston at Cambridge, we've kind of done a model where you can behaviorally push these distributions closer together or further apart to try and homogenize your labels, which produces some interesting predictions that we want to test. So basically, when you and the opposing group are of intermediate dissimilarity, then you should get maximum homogenization effort. And in that grant, we're going to try and take that model and test it with Barbara Caspers and Hazel Nichols and Zav Harrison by analyzing allo marking before and after these conflicts and looking for homogenization. We're also going to trace longitudinally the kind of anal pouch microbiome, which we think is the source of this individual identity. Right, I'm just going to very quickly have some speculations about the cognitive dimension of identity. You know, some interesting ideas in the psychology literature, the idea that in times of war or in extreme duress, there's what's known as identity fusion. Humans undergo identity fusion, which is this visceral sense of oneness with the group and the kind of merging of the self with that of the group. And that's been used to explain these crazy acts of self-sacrifice in war, running towards machine gun encampments and so on. And it's distinct really from these kind of ideas about what makes humans unique cognitively in terms of cooperation, the idea of sharing intentions and sharing plans and so on, which has been stressed by some researchers. It's more just a subsumation of the individual's identity. And I like this kind of idea, first of all, because the mongooses, I can believe that when the enemies appearing in front of them, they don't care about anything else other than fighting. You know, they're not aware of you. They're no longer, you know, they're not aware of each other. They're not competing. They're just, for that moment in time, they're kind of fused. And this fits with this interesting, slightly out there theory, I think, away from the mainstream in economic game theory, the idea that humans have the ability to flip into a different cognitive frame. So instead of asking themselves, what should I do in any particular game situation, they have the capacity to flip into this we mode asking instead, what should we do? It's an alternative model of rationality. And Bacharach, this book that I was very influenced by Michael Bacharach, suggests that this capacity has evolved by strong intergroup competition between group selection. But it's a sort of idea that doesn't require much complexity in terms of sharing intentions and joint commitment and so on. So I think it could apply to things like wasps and mongooses quite well, actually. So that's something that I would like to explore in future. And the final thing I'm going to say is that in mongooses, your identity can switch pretty quickly. So this is from a paper I wrote from my PhD, where I say that there was a female group occupying an area, and they were kind of fighting with their neighbors, and particularly this one big group. And we were watching them fight, and then I went away thinking that they'd gone into the bush. It was 12 o'clock. I thought, okay, nothing's going to happen. So I came back at 4 o'clock, and a whole load of males had left and joined that group and were fighting against their natal group, really vicious fighting against the group that they'd been born into and spent their whole lives in up until that point. So something had changed cognitively. They'd cognitively switched identities to these other groups. So I hope I've convinced you that there is something interesting to explore using animal societies about these two other aspects of major transitions, which allow this interesting phenomenon of unity while retaining diversity. And that's it. Thank you for listening.